speak about the activities that we are doing within the FAIRification project. So here is the latest reached the topic to preclinical image dataset. And essentially, there are, I would say, two main limitations. One related to the complexity when you deal with preclinical data set that is, let's say, a multi-domain and multi-level field, I would say, because of there are a lot of instrumentation that can be exploited to obtain preclinical images. You can obtain different type of raw images from, let's say, anatomical to functional, or providing metabolic information. You also the study protocol and experimental procedures are usually quite different across different studies. And when you start doing some processing, you can also exploit different type of processing tools, obtaining different parametric images, again, keeping different type of information and obtaining different type of output following the analysis. So one of the main difficulty and complexity is how we can track all the essential information that we might need to describe a preclinical data set. In addition to, let's say, generating preclinical images, there is another limitation that is where I can find preclinical image data set. Main reason is because of, in contrast to the, let's say, biological image feed, there are nowadays there are very few FMNT initiatives, usually at a local or single site institution level. Even if there are some repositories, some of them can lack of metadata information or they are not curated at all. Some of them are limited to specific domain. And some repositories might have only very small data sets or that are not completely free or accessible. And here you can see in the slide some example of repositories for preclinical data sets. So those are, of course, limitations that hamper the sharing and even the fundability of preclinical image dataset. And hopefully the FAIRification project wants to, let's say, provide some tools, some guidance, recommendation in order to improve data sharing. And at least I will talk more about the sharing for preclinical open images, as well as implementing the FAIR principle for the preclinical data. And so we'll talk mostly about the metadata and the ontologies. In fact, if you look to what you really need in order to improve the sharing of data, essentially you need two things. The first one is an harmonized and structured metadata model. So meaning essentially a list of metadata, so a list of information that describe from several perspectives the images that have been acquired. And on the other hand, you also need to exploit some ontologies in order to curate the repository, so in order to make the metadata more findable and more interoperable. So what we did the foundingGIDE project, and actually these are some activities that we started a few years ago within the working group of biomedical image data within the Euro-BioImaging research infrastructure, was to start developing a structured metadata model by taking advantage of other and similar initiative and recommendation, including the RMB model that we already heard during these 2 days, as well as taking advantage of recommendation from the ARRIVE and the PREPARE guidelines, as well as also look into some standardization guidelines to describe, for example, method description for preclinical imaging. So starting from this previous work, we have structured a metadata list to describe preclinical image dataset that is based on 8 main sections. Some of them including additional subsections that essentially provide information regarding the study design, so general information on the study and the associated publication, the study component, so meaning related to the imaging modality and to the imaging facility where images have been acquired, and as well as licenses to reuse the data set. We are taking information about the in vivo experimental parameters, so all related to the in vivo study, as well as to the experimental procedures. So any information related to any pharmacological, surgical, contrast agent administration, histological procedures, and so on. And then some sections that are more related to image acquisition, so instrument vendor, specification related to the image acquisition, image data information, imaging correlation if there are more than one imaging modality, and eventually for co-registration of images across multiple modalities, and then information related to the way in which the data have been analyzed. And so all of these sections essentially provide a full picture of the preclinical image dataset. You can see here some representative example providing all the different sections and all the metadata that are collected. So starting from the study type, study design. Of course, if we are talking with the preclinical data, usually they are in vivo data. All information associated to the publication that is linked to the image dataset. Information related to the study component. Okay, and so related to the imaging modality, image submodality, image coverage, image target, dataset information, so related where the images have been acquired, in vivo experimental parameters, and so you can see here all the different, for example, cohort of mice that have been injected, which type of disease model, which type of cancer model, species, strain, and so on. And then you also have all the experimental procedures, for example, which type of molecule have been injected. In that case, they are actually contrast agent, which type of cell line have been used, and so on. And then information, for example, on image acquisition, and so instrument and vendor, instrument type specifics, information about the images, and information related to the analyzed data. So as you can see, there is a quite a full list. We have almost 200 metadata list that can describe all of this dataset. And very importantly, also we included the information related to the way in which you can reuse the images. And so we have metadata that describe the license type that is associated to the image dataset, as well as ontologies that describe how you can reuse the data. And so here you can use the exploitation of the data use ontology that provide information, data use permission, in which ways you are allowed to exploit these images, as well as a description of the type of investigation. And so in that way, person that might be interested to reuse the data, they know if they are allowed and which way they can exploit these images. So one hand is related to the metadata, and in order to reach a consensus on the metadata, the aim is also to we are developing a survey in order to reach a consensus on this metadata list, with the aim to touch other preclinical imaging communities worldwide. Of course, the larger will be the answer from the survey, the larger will be the consensus on this metadata list, where we would like to identify recommended and optional metadata to describe the images, as well as to reach a consensus on a minimum set of metadata that could be exploited to describe instrument specification per each imaging modality, as well as image acquisition parameters per each modality. And if you are interested to participate in the survey, please send us an email, since in a couple of months we will be able to start the survey procedure. Another part that is related to the metadata description is also the concept of ambiguity. This means that usually when you use some term to describe, let's say, a molecule, to describe a concept, maybe you can use one term and another researcher might use another term. And this means that if you are looking for specific images, it could be that you are not able to find some images accordingly to the query or to the term that you are using when you are performing your query. As common example is we are looking, for example, for images that are associated to breast cancer. And if you have labeled your images with the breast cancer, but you are performing the query with the term mammary cancer, likely you will not be able to find the images. And this means that you need a way to resolve this ambiguity. And the way that you can exploit is to use the ontologies that essentially you can consider an ontology as a kind of a vocabulary having inside several terms, and within the vocabulary you also have all the relation, the concept that allow you to make this relationship and to understand that when you are referring to a breast cancer or to a mammary cancer, essentially you are referring to the same, let's say, disease. And so in that way you will be able to find the same images independently of the term that you are using within your query. So because of also the preclinical imaging field is a multi-domain field, what we did within the project was to identify several domains that can be exploited to describe preclinical image datasets. We essentially identify 14 different domains, and for each domain we look to the ontologies that can be exploited to describe each of these domains. And so you can see here all the different domains and the list of the ontologies that we have identified that can be used to describe each of these domains. And then we look into each ontology by looking to different, let's say, properties, including the file format to which this ontology is available, the number of terms of each ontology, essentially meaning that the size of the dictionary, if the ontology is freely usable or not. We look also if the ontology is maintained, so look into the update of the ontology, to the, let's say, usage of the ontology, how much is used the ontology within each domain, and so on. And so by extracting all of this information, then we developing a kind of a scoring metric in order to score all the ontologies inside each domain, and in that way, in order to identify, let's say, the potential best ontology per each domain. And this is what we call the landscape analysis in order to identify a specific ontology per each domain. And here you can see the results of this scoring process. And here is the list of each of the ontology that we have selected and that we will use in order to describe specific metadata and in particular specific term per each domain. And hopefully in that way we will improve the findability of the preclinical image dataset. And so having these two, let's say, steps almost done, then we will exploit the metadata list and the ontology in order to develop and to build a preclinical image dataset repository. So far we have started by developing what we call the PADAR. The PDAR, that is a repository for metadata that describe preclinical image dataset that are associated to peer review publication. So we started a couple of years ago by developing the repository. So far we have 19 datasets that have already been deposited. The repository can cover multimodality images. It's we'll say a well-curated repository with almost 200 metadata covering different feeds, and as well exploiting also ontology in order to resolve the ambiguity issues that I described before. Some of the preclinical datasets have also information related to the images that can be hosted in different repositories. We also have our XNAT instance where we can host some of the datasets that are listed here. And if you want to have more information, you can look to the QR code that is provided here. You can also contribute if you want. You just go to the submit page of the PIDA repository. You can find here the template that you can fill by looking to the submission guideline, and then after you fill the template, then you can send us the metadata template, and then we will upload the dataset to the repository. And with this, I would like to thank all the people that contributed to these activities. First, the working group on data management for biomedical data within the Euro-BioImaging infrastructure, all the funding and partners that are following and helping by providing recommendation on the metadata list and the ontologies and so on, as well as all the MEDAB team and the PIDA development team that is working on developing this repository. And thank you for your kind attention.